everyone, and welcome to today's webinar, Epithelium Formation, Cellular Adhesion Regulation in 3D. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Leica Microsystems. To learn more, please visit leica-microsystems.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. And with that, I'd like to now welcome our speaker, Dr. Rafael Garcia-Mata, Professor, Department of Biological Sciences at the University of Toledo. For a complete biography on our speaker, please click on the Presenter tab. Dr. Garcia-Mata, you may now begin your presentation.
Well, thank you very much for this introduction. Before starting, I also want to thank Leica for the invitation to present at this webinar series. I'm going to talk about today some of my work related to work that we've done in relation to the architecture of the epithelial cells. My lab is interested in understanding the mechanisms of regulation of the actin cytoskeleton. And as you all know, the actin cytoskeleton controls a wide range of cellular processes in the cell, which includes cell adhesion, cell migration, cell invasion, polarity, cytokinesis, and many, many other processes in the cell. And as you all probably know, actin can take several different shapes and structures in the cell, and it's also a very dynamic structure. So it can change structure and shape, and it can modulate cells dynamics and cell contractility. So it's actually one of the key processes in regular cell adhesion and migration, which is some of the things that my lab is interested in understanding. Several years ago, the seminal work from Alan Hall and others showed that the regulation of these actin structures is mediated by a family of proteins called the Rho family of GTPases. And what the Hall lab and many other labs shown is that if you modulate the activity of these GTPases, you can also change some of these cellular behaviors that have been observed. For example, if you modulate the activity of a Rho protein called Rho-A, what you see is that you increase the number of stress fibers and contractility and focal adhesions. In contrast, if you activate another Rho GTPase called RAC, what you do is you stimulate cell spreading and migration. And then if you stimulate CDC42, another Rho GTPase, you stimulate the formation of filopodia. So my lab has been interested in studying the mechanisms of regulation of Rho GTPases for many years. This family of protein comprise 20 different members, and within those you will see the ones that I just mentioned, Rho-A involving contractility, RAC involved in cell spreading and migration, and CDC42 protein involved in filopodia and some other things. Now, Rho proteins function as molecular switches, and that means that they can exist in two different states: an active state, which is bound to the nucleotide GTP, and an inactive state that is bound to GDP. The interconversion of these two states is regulated by two additional family of proteins, the Rho-GEFs, or one-in nucleotide exchange factors. They are the ones that promote the incorporation of GTP to activate the protein and the release of GDP to inactivate it. There's another family of proteins called GAPs, or GTPase activating proteins, that regulates the hydrolysis of the GTP to inactivate the proteins. When the proteins are in the active state, they interact with effector proteins, which will typically regulate a particular signaling pathway. Now, The Rho GTPase, these 20 members of the Rho GTPase families are regulated by a much larger number of upstream regulators, including 80 Rho GEFs and 66 Rho GAPs. So this poses a particular problem or challenge, if you will, when you're trying to understand this. And I'm just giving you one example here. For example, if you have 3 GTPases, the ones that I mentioned, you have a much larger number of Rho GEFs that can activate them. And then to complicate this a little bit further, you have some Rho GEFs that can activate more than one GTPase. And then, of course, when you're looking at the downstream effectors, you have almost 100 effectors that can be activated by different Rho GTPases. And again, the same problem, some Rho GTPases can activate more than one effector. So one of the questions that my lab is trying to understand, or one of the challenges in the field, is how does the cell decide, for example, when a GEF is stimulated to activate, say, this particular pathway as opposed to this particular pathway? And this is one of the questions that my lab has been interested in understanding for many years. And we try to understand different questions related to this problem. For example, what are the functions of these different Rho GTPases, these 20 genes in the cell? What are the upstream and the downstream regulators? What GEFs and GAPs and GTPase and effector combination is activated for each of the different pathways? And then when are these pathways activated and where do they operate in the cell? And of course, what are the molecular mechanisms controlling its function? So many years ago, I started looking into this problem. And what I was trying to do is I was trying to understand whether there are some general mechanisms that you can find that maybe regulate, so general principles of regulation of these proteins. And I'm going to show one example here. This is the family of Rho GEFs, these upstream activators of the Rho GTPases. They're very diverse. They have different domain structure. But one thing that I found several years ago, that many of them have a similar characteristic. And this characteristic is at the C-terminal tail. They can bind, they have a sequence called PDZ binding motif. It's a sequence at the C-terminal tail that allows them to bind to a domain in other proteins called PDZ domains. And these PDZ domains are very abundant. They're usually, encoded in more than 350 proteins, and they're usually represented more than once in a single protein. So they're typically present in proteins that are called scaffolding proteins, proteins that are recruit and bring particular proteins to particular sites of the cell through protein-protein interaction. So the thinking then was, okay, these GEFs may be interacting with PDZ proteins. These PDZ proteins are going to localize these GEFs at the right time of the cell, at the right part of the cell, at the right time to activate a specific Rho GTPase at a particular time locally to regulate a specific signaling pathway. Sort of that's the theory, and that's what we've been trying to understand in the past few years. Today, I'm going to tell you the story about one GEF that through this interaction, it's targeted to the right place, to the right time, and regulates a particular pathway. And the GEF that I'm going to introduce today is called SGEF. It does have this PDZ binding tail that I mentioned about. And the way we sort of went about trying to understand this is first we did proteomics and some other assays to try to identify protein interactors for this particular GEF. And what happens is we found two proteins that interacts with these GEFs. So before I show you the identity of these proteins, I will tell you a little bit more about this GEF. This GEF is specific for a Rho GTPase called Rho G. Rho G is similar to one of the ones that I mentioned, RAC. So it's important for cell spreading, cell migration, and some other functions in the cell. As I mentioned, it does encode this PDZ binding tail, which is a similar characteristic of all these other GEFs that I described a minute ago. And we found it interacting with two proteins, one protein called Scribble and the other one called DLG. These two proteins have multiple PDG domains, so potentially they can be interacting with this domain in the cell. And the thing that caught our attention about these two proteins is that these two proteins function in same molecular complex, and they perform a very important function in epithelial cells. What these two proteins do is they are part of what is called the scribble polarity complex. So I'm going to take 1 minute to introduce what this complex does. So this complex is important for the establishment of cell polarity in epithelial cells. So what is a cell polarity? So as you probably know, epithelial cells are cells that are very important in tissues and organs to establish a barrier. So they typically separate the outside from the inside. And they do so by establishing what's called a polarized phenotype, in which the apical surface, which is what is facing the outside, is separated from the basolateral surface, which is what's facing the insides of the tissue or organs. And this is mediated by the formation of cell-cell junctions, called adherence junctions, and tight junctions, which form a barrier that does not allow molecules and solids to go through in between cells. So this barrier function is very important for protection, to prevent infection, to prevent leakage of the tissues. So the establishment of this cell layer of epithelial cells is of crucial importance for the development of tissues and organs. Now, the formation of this very organized architecture depends on these protein complexes. Actually, 3 protein complexes that work in synchrony. One is called CRAMS, the other one is called the PAR complex, and the other one is called the scribble complex. They all work in synchrony to establish this particular cell architecture. Now, the fact that we found a row GEF that is interacting with 2 members of the scribble complex really caught our attention because of the importance of this complex on developing polarity. So our goal is to try to understand why is this GEF interacting with the members of the polarity complex. So our initial idea was, okay, this binding motif is going to interact with one of these PDG domains in either one of the proteins, and we'll go from there. We'll see what they do in the cell. So first, I'm going to summarize some of the results that we already published. And then I'm going to, in the second part of the talk, I'm going to talk about more of our new and published results. So in this slide, I'm going to summarize a lot of results that if you're interested, you can please look at the publication listed at the bottom, and you can see more details. So this is what we found. We found that SGEF binds to scribble, and it binds through this, a new sort of, instead of binding through the tail, as we predicted, it binds through a new internal PDG binding motif. Since this was a new binding domain, we characterized it, and we even got the crystal structure of it. So DLG also binds to the N-terminus of SGEF, and not through the PDG domains, but through a domain called the GOT domain. The interesting also finding in our previous publication is that these 3 proteins can bind simultaneously, forming a ternary complex. So SGEF forms as a bridge, brings these 2 molecules together, and they form this ternary complex. This is relevant because a lot of people have studied the scribble complex, and they always found that even though they perform a similar function and they are required for the functions of the scribble complex as a whole, they do not interact physically with each other. So what we found is that SGEF brings these 2 proteins together. Now the question is, what do they do? Why is this important? Our idea is that GEFs needs to be localized at the right place and at the right time. So we predicted that this complex is going to bring SGEF maybe to the basolateral membrane, where it may play a role during either cell junction formation, the establishment of polarity, or maybe also the activation of rho G and the activity of rho G downstream regulated some processes that we still don't know. So we went on to try to study this, and we did it. We started in a very simplistic way. What happens if we knock down the expression of SGEF? And what happens? And we looked in two different places. We looked at the tight junctions level, essentially at the level of the barrier function, and then at the cell-cell adhesion level, at the level of adherence junction, to see if any of these processes are affected in the absence of our gene. So we did knock down, and then we stain. We do all this work in MDCK cells, which is a very nice cell line from canine origin, which has been used extensively to study cell-cell adhesion and polarity. So if we stain these cells with a tight junction marker here called Z01, we see very nice the cell-cell adhesion borders stained with this nice antibody. When we knock down SGEF, we instantly saw a phenotype in tight junctions. And I hope you can appreciate here that the junctions, instead of being curvilinear, which is very common in MDCK cells, you see now that they're very, very straight. Many times when you do a knockdown experiment, you want to make sure that there's not off-target effects. So the one way to rule that out is to rescue the expression of your gene and see if it rescues the phenotype. And this is what we did. We re-express a fluorescently tagged SGEF, and you can see how we rescue the curvilinear phenotype of the knockdown. So what are all these, what do these linear junctions represent? So these linear junctions may indicate that these junctions are now under tension. Most of these cell-cell adhesions are connected to the actin cytoskeleton. The actin cytoskeleton can contract through the actomyosin system. And one thing that this phenotype indicates is that this may be contracting and exerting tension over the junction borders. So we tested that by looking at myosin. I'm going to skip this for a moment, but essentially this is only an assay in which we quantitated the curvilinear aspect of the junctions, and we see that it's affected. And what we also measure is the permeability of the junctions using this assay called transepithelial electrical resistance. And what you can see here is that when we knock down SGEF, the barrier function is impaired, meaning the resistance is lower, and this indicates that the junctions are more permeable. So when we looked at the junctions in a little bit more detail, what we see, if we look at myosin, now we're looking at the contractile machinery, we see now that myosin is recruited to the junctions in this very nice sort of periodic pattern, suggesting now that these units are much more contractile than the curvilinear ones, and they sort of they are exerting tension over the junctions, and this is why they are linear. So for the rest of the talk, I'm not going to talk about the tight junction phenotype. This is something that is very interesting, and we're very interested in continue exploring it. But for the rest of the talk, I'm going to talk more about the phenotype on the adherence junction. So the phenotype on the adherent junctions is the following. So I show you this linear phenotype at this level, at the tight junction level. Now I'm going to show you a staining for E-coherine, the molecule that functions to attach one cell to the next by forming these homophilic interactions. So when we stain for E-coherine in the knockdown cells, we saw something very striking. And the striking phenotype is the E-coherine staining is almost completely gone. You can see how E-coherine is in the junction in control cells. When you look at the SGEF knockdowns, the E-coherine levels are almost non-detectable. Now, again, we do the same experiment. If we rescue with the expression of SGEF, what we can see now is, for example, in this little island that is re-expressed in SGEF, we see how E-coherine is very nicely restored, only in the cells that re-express SGEF. In the cells that are not re-expressed in SGEF, it's not restored. So this is very specific to the expression of the SGEF protein. So what does that mean? The other thing that we did is we look at the expression levels by Western blotting. So what we want to know is whether E-coherine is redistributed out of the junctions into the cytosol and this is why you don't see it, or if actually the levels of the protein are downregulated in the cell, and this is what actually happens, the level of the protein are downregulated in the cell, as you can see in this Western blot. The other members of the complex, DLG and Sztul, are fine. We don't see a change on the expressions in those. Now, why is this important? Why is Ikahiren? Ikahiren downregulation is actually very important because it's one of the earliest hallmarks of cancer. So a couple of things that are important. First, 85% of the cancers are of the epithelial origin, which is the cells that we're studying. And then a lot of these cancers start at the very early stages by downregulated Ikahiren levels. And this, what happens when Ikahiren is downregulated is that junctions are disassembled, and then now the epithelial cells will lose the monolayer organization. They can now proliferate more, and they can also invade more. So this is one of the early signs of cancer, when the epithelial monolayer is disorganized and proliferates more. And typically this is initiated by a loss of Ikahiren cell. So there's a lot of interest in trying to understand what are the mechanisms of Ikahiren downregulation, because this can shed light into how we understand the early progression of cancer. Now, the questions that we want to understand is how does this complex of these three proteins, with a protein that has an activity to activate a particular rho GTPase, how is this connected with regulating the stability of Ikahiren, and how, when we impair the formation of this complex, now Ikahiren is downregulated or degraded? So we have a couple of questions that we're going to explore today. The first one is, what is the role of the other two members? So do they have the same role that SGEF? What is the role of the catalytic activity? Is activating SGEF part of this process? And then, how is this process regulated? How do you go from here to here? So we're going to explore some of these questions in the rest of the talk. Okay, so the first experiment that we did is we knock out the expression of the two other members. As you can see here, we knock out the expression of Sztul knockout cells. We knock out the expression of DLG knockout cells using CRISPR technology, and then we analyze the phenotype. And the question is, does knocking out these other two genes also promote the downregulation of Ikahiren? And the answer is not really. What you can see here by immunofluorescence is when we knock down SGEF, we see a severe downregulation of Ikahiren. When you knock out the other two genes, you see some redistribution, but not as drastic as the one with SGEF. And if we look at this by Western blot, we see that the downregulation of Ikahiren is slight, but far from being as severe of knocking down SGEF. So the bottom line here is not the same to target each of the members of the complex. Targeting SGEF is a little more severe than targeting the two other members. The next question that we wanted to ask is whether the activity of SGEF that is activating this downstream rho GTPase called rho G is required for that. So I show you this experiment. We can knock down SGEF, and we can rescue by restoring the expression of a fluorescently tagged SGEF. So to analyze the role of the activity, what we did is we did another rescue experiment in which we rescue with SGEF, but with a construct of SGEF that is mutated in the catalytic domain. So it's called what's called catalytic dead. So it cannot activate rho G. Everything else in the molecule is the same, but it doesn't have catalytic activity. So if the catalytic activity is important, then this construct shouldn't be able to rescue the expression of Ikahiren. And it doesn't. So the answer is yes, the catalytic activity is important, because when we put this construct, we cannot rescue the expression of Ikahiren. So we took this a little bit further because we want to make sure that the catalytic activity is important. And we also want to sort of distinguish between two possibilities. What I show you so far is the following. If we knock down SGEF, then Rho-G cannot get activated, and now E coherent gets degraded. I also show you that if we put a catalytic del mutant, we cannot rescue. So now we designed an experiment to test two other things. One is, if we put just the catalytic activity alone, not targeted to the right place, it potentially can activate Rho-G. Will this be enough to prevent the degradation? And then we did another construct that is artificially targeted to the basolateral membrane, independently of the complex. So the question here is, if we put the activity at the right place, is that enough to rescue E-coherent activity? Now the answer is yes, you need the activity, and yes, you need the activity targeted to the right place. You can see here in the Western blot that the construct that target the activity to the basolateral membrane rescues the expression, but the catalytic activity alone, very, very little rescue compared to the targeted one. We can see the same thing by immunofluorescence here, where we see rescue of E-coherent only with the targeted activity, but not with the cytosolic activity. So as a summary of these studies, the activity is important, and the activity localized at the right place is also important. Now, how is E-coherent downregulated in these cells? I told you that we need the SGEF, and I told you that we need the activity. I also told you that E-coherent is so important for cancer progression that there's hundreds and hundreds of papers of E-coherent downregulation, and people have found that there's many different ways to downregulate E-coherent activity. And this sort of cartoon summarizes some of these studies. So what people have shown is that when E-coherent is engaged and interacting with another cell, it's very stable at the membrane, and it forms these sort of clustered molecules of E-coherent that are interacting with the other cell, and they're very stable. But there's also an exchange between stable and unstable E-coherent, and when E-coherent is destabilized, it can get endocytosed. And it can get endocytosed through different mechanisms, through class III mediated endocytose or caveolin mediated endocytose. Both of these coherents can go to endosomes, and they can either end up in the lysosome and being degraded. They can be degraded by the proteasome, or they can also be recycled back into the plasma membrane. So there are many ways to control how much E-coherent you have at the membrane, mostly by internalizing or either sending it to degradation or recycling it back to the membrane. So we decided to test some of these possibilities to see what happened when we knock down SGEF. What are the systems that are being utilized to downregulate E-coherent? The first one that we tried was protein degradation. So what we did is we inhibited both the lysosome and the proteasome using different inhibitors, MG132 for the proteasome, chloroquine for the lysosome. And what we can see is a partial rescue when we inhibit the proteasome and no rescue with the lysosome. The rescue is still very poor if you compare the controls of the treated cells with the control cells. It's still not even rescued to 50% of the levels. So this suggests that degradation by the proteasome may be important, but it doesn't explain the whole picture because we cannot rescue all the levels of E-coherent in control cells. So we decided to test a couple of other systems. So I'm going to go back to this diagram that I showed before. And one of the things that people know in these early stages of cancer development when E-coherent is downregulated, that in many cases E-coherent is downregulated at the transcription level. And this is mediated by a family of transcription factors. They're actually transcriptional repressors, or a family called SNAIL. And this SNAIL family has many members, including SNAIL as SLAG, and they mediate this process called epithelial to mesenchymal transition. And this is when cells lose the epithelial identity, this polarized epithelial identity, and then they become mesenchymal, more invasive, more proliferative. And this is mediated by these factors that regulate, actually repress the transcription of ikahirin. So we wanted to see if transcription was involved. So what we did first is we did qPCR and analyzed the messenger levels of ikahirin in control and in SGEF knockdown cells. And what we saw is that transcription was severely impaired. So we see a lot less mRNA for ikahirin when we knock down SGEF. As we've done before, we showed that we can rescue this by putting the wild type SGEF, and also that we cannot rescue it with the catalytic dead. So this also depends on the catalytic activity. So the bottom line is SGEF can modulate the transcription levels of ikahirin, and this also requires the catalytic activity. Now, we looked at these transcription repressors that I just mentioned, snail, SLAG, and some others, and what we found that one of them, SLAG, was drastically upregulated when we knock down SGEF. You can see here how ikahirin goes down and SLAG goes up, and this is the protein that will repress the transcription of ikahirin. To make sure that SLAG was the factor that was actually responsible for the downregulating ikahirin, we did this following experiment. What we did is we silenced the expression of SLAG in this cell line. So we make double knockdown cells that are SGEF knockdown and SLAG knockdown cells. And if SLAG is responsible for downregulating ikahirin, if we silence SLAG, then we should rescue ikahirin expression. And this is what we see. This is the experiment. If we silence SLAG, we should rescue ikahirin expression. And this is what we see. We see ikahirin rescue here, and we also see the efficiency of the SLAG knockdown. If we look at cells, we see the same thing. We see how ikahirin is nicely rescued. But we also see something interesting. If you look, this is the side view of the cell. If you look at the side view of a control cell, you see how ikahirin is nicely localized in the lateral membrane. Interestingly, in the SLAG cells, it's localized at the lateral membrane, but also at the basal membrane. And what this suggests to me is that even though you can rescue the levels of ikahirin at the Western blot level or at the protein level, the localization of ikahirin is still affected. So since we still don't have this ternary complex that I've been telling you about, what we propose is that this complex is important also to regulate the right localization and the stability of ikahirin at the membrane. And I will show some experiments in a minute or two. Now, we also tested the activity, so we're sort of building up this sort of hierarchy of the pathway. So we want to know now if this upregulation of SLAG depends on the activity. As we did before, we can rescue it with the wild type SGEF. We see how SLAG goes down when we put back SGEF. If we put back the catalytic dead, now the same thing, we can rescue the SLAG levels. So again, everything that I show you so far depends on the activity of SGEF. And presumably the activation of ROG. All right, so how is SLAG upregulated? So I'm keep going sort of up and up, trying to connect ikahirin and SGEF to the transcription regulation of ikahirin. One of the things that I did not mention in the introduction is that ikahirin at the membrane typically exists as a complex between ikahirin and the catenin proteins, mostly alpha-beta-catenin and P120-catenin, which associate to the tail of ikahirin, stabilize this complex, and connect it to the actin cytoskeleton. This complex is also very dynamic. It can be regulated by phosphorylation and by some other means. And when this complex is dissociated, there's a couple of things that happen. One, ikahirin is less stable, and it can be internalized, and I'm going to show this later. The other thing is beta-catenin is released into the cytosol. It can translocate into the nucleus, and it can also regulate gene expression. And ikahirin, beta-catenin is known to be one regulator of the expression of the protein SLAG that I've been mentioning in the past slide. So we wanted to test if beta-catenin was responsible for regulating SLAG. The way we did it is there's an inhibitor that inhibits beta-catenin transcription activity. So when we treat these cells with an inhibitor called ICRIT, what we see is that we rescue ECA levels very, very efficiently. So as I'm putting sort of systematically building this pathway, we know now that beta-catenin can regulate the expression of ECA at the transcription level, probably by upregulating SLAG. And we tested that by looking at SLAG, and we see that when we inhibit beta-catenin, the increase in SLAG levels is restored. So it's essentially going back to normal. So the sort of the pathway goes from beta-catenin, translocates to the nucleus, activates SLAG, SLAG inhibits ECA, and this is all dependent on the presence of SGEF and its ability to activate ROG. Okay, so now, how is ECA downregulated? So we keep sort of hammering at this protein. I show you that beta-catenin is probably responsible for activating SLAG. Now, how do you regulate those sort of this stability? So for ECA, so for this complex to be dissociated, it probably needs to be phosphorylated or dissociated some other way. So one thing that we can do is we can test for the presence or absence of this complex in the cell. And we can do this using immunoprecipitation. We can immunoprecipitate, for example, P120 and see if ECA binds, as we can see here in control cells. If we do this in SGEF knockdown cells, the complex is not there. And the opposite is true if we immunoprecipitate ECA and we blood for P120, we don't see P120 in the complex. So this suggests us that when we don't have SGEF, this complex is dissociated and also means that ECA is less stable. Now, how can we test for ECA stability at the membrane? We can do this using a microscopy assay or microscopy technique called FRAP, or fluorescence recovery after photobleaching. And what we do, the way we do this for ECA is we have cells that are expressing GFP ECA, control, and SGEF knockdown cells, and then we bleach a region of the junction using a high-intensity laser, and then we measure the recovery of the fluorescence into the bleach area. And that typically gives you an idea of the diffusion rate of the proteins in the membrane and the recovery of the fluorescent by other means. In this case, it could be by newly synthesized protein that comes into the junctions or by endocytosis. And the prediction is if ECA is less stable in SGEF knockdown cell, it should diffuse a lot faster. So the recovery should be faster because we know that ECA is very stable when it's in complex with ECA on the other cells, and the recovery is a lot slower when we bleach an area. So how do we quantify this? This is an example of one of the bleach areas in control and knockdown cells. You can see a junction. You can see the square in which we bleach the fluorescence. And then you can see after bleaching the area how the fluorescence is recovering over time. So this is just one snapshot of each of the different stages. We can measure this or we can visualize this using a kymograph in which you can see the sort of the fluorescence intensity along a junction is this axis, and then you can see time in this axis. So here you can see the bleach event and the bleach area, and how over time in control cells, it takes quite a long time to recover. You see that the bleach area is very stable, meaning that there's no fluorescence coming in from the sides, and then very, very slowly it starts to recover. In contrast, what we see in SGEF knockdown cells is the bleach area is not as stable, so it starts to get recovered a lot faster, and then you recover the fluorescence in just a much shorter time, suggesting again that diffusion is a lot faster. We can graph this over multiple bleaching events, and what we can see here is that if we measure the half-life, meaning the 50% of recovery of fluorescence, you see that SGEF knockdown cells recover in half the time, so twice as fast, sort of confirming that the stability is one of the things that is affected when we knock down SGEF. All right. So the final experiment for this part is, if this is true, EK-hearing is unstable, then it should be internalized faster. And if we were able to inhibit endocytosis, we should be able to rescue this phenotype. And this is exactly what we tried, and it also worked. If we inhibit clathrin-mediated endocytosis with an inhibitor called Dinosaur, we rescue EK-hearing very nicely. If we inhibit with a non-clathrin-mediated inhibitor, we have some rescue, but not as good as with the clathrin. As you can see here, clathrin rescues very nicely, almost to control levels. The other thing that the clathrin inhibitor does is that it rescues lag expression very nicely. So I think that these results suggest that this SGEF is regulating the stability of EK-hearing and also the clathrin-mediated internalization and eventually degradation, which potentially will release beta-catenin. So to summarize this part, we show that this ternary complex composed of SGEF, scribble, and DLG through the activation of Rho-G regulates the stability of EK-hearing. EK-hearing gets internalized and potentially degraded, which also releases beta-catenin, which then regulates the transcription of SLAG, and SLAG is a transcriptional repressor of EK-hearing. So this is a feedback loop that eventually results in the downregulation of EK-hearing. I'm going to shift gears for a moment for the last few slides to talk about what happens in 3D. So the MCK system is a very nice system to study sort of the early development of an organ. And as you know, organs are usually composed of tubules and lumens, and these cavities are also surrounded by a layer of polarized cells. So you can sort of recreate the early stages of this with these cells if you play them in a 3D matrix. And if you play them as single cells, they eventually will divide and they will form a cyst or a spheroid in which you have a layer of epithelial cells that are perfectly polarized with the apical surface facing the lumen and the basolateral surface facing the outside. So this is a very nice model to study polarity, because when the cells are wrongly, or so there's defects in polarization, the cyst is not going to look like this. So I'm going to show you some examples. So we very nicely can recreate this in our lab. We can see, for example, in control cells, we make these very nice cysts. Where if we stain for a marker of the apical membrane, it's very nicely localized in the center of the cyst. We can stain for beta-catenin in this case. Here in junctions, you can see the junctions between cells. So very nicely, a very nice model to study this. Now, what happens when we knock down our gene? What we see is a very, very drastic phenotype. We see that the polarity and the organization of the cyst is affected. We see these sort of patches of lumens in the center of the cell. Some of them seem to be open. Most of them seem to be closed. So there's a really disorganized cyst in the end. We can rescue this if we put back our wild type SGEF. We go back to single open lumen. We can also quantitate this, and we show that most of the cysts in control have open lumens. Very few of the SGEF knockdown cells have open lumens, and then we can rescue pretty nicely these if we put back wild type SGEF. So what happens to the other members of the complex? If we knock out the two other members of the complex, we also have severe defects in cell polarity and lumen organization. You can see that both scribble knockout and DLG knockout have also multiple lumens, also open and closed. And actually the most severe phenotype is the one of the DLG knockout cells. So the questions that we're trying to understand now is, how do you get from here to here? So how do you make a multi-lumen cyst that is completely disorganized? And there are many different ways to make a multi-lumen cyst, mostly through defects in cell and vesicle traffic or cell division, or cell-cell adhesion. So this is what we're trying to explore now. For that, we need to build a lot of tools. And this is where also microscopy is helping us quite a bit. So I show you the control and escape knockdown cells, severely disorganized. So what we can do is we can make 3D stacks of these cysts, and we can sort of render them in 3D in an imaging software, and then we can measure the different parameters of the cyst. And I'll show you some of the things that we've been measuring. We've been measuring cyst volume. As you can see here, escape knockdown cells, cyst seems to be larger, and this is confirmed when we measure volume. We can also measure E-coherent expression, the same as in 2D. E-coherent is severely downregulated in the 3D cysts. Then we can also measure the number of lumens. We can reconstruct these lumens in 3D and then measure their number and also their volume and some of the shape descriptors too. So this is something that we're doing now. Now, the thing that I'm also most interested in measuring is, hopefully this movie will play. Okay, we'll wait a little bit as it sort of renders. Okay, there it goes. So one of the things that we've been doing is trying to follow the cyst formation from a single cell to a mature cyst in live cells in 3D. So we have the cells making cysts. In this case, they're labeled with a membrane marker, and we follow them over time, and we make 3D stacks at different time points, and then we can make a time lapse, and we can follow the cyst development over, in this case, 4 days under the microscope. And the goal for this is that we can detect the moment when these defects start appearing. Do they appear very early during the cyst development? Does it involve cell division? Does it involve junction or movement of the cells in a particular way? So by imaging these with different markers, we're going to be able to dissect that. And I'm going to show you 1 or 2 examples. So this is one example. I show you how the normal cyst, they divide and they rotate and they form a normal cyst over 4 days. I'll show you what happens when we knock down SCF. So you're going to notice when this movie starts playing that these cysts don't behave the same way. First, they move a lot. They seem to be migrating very actively. Second, they fuse to each other. You're going to see now this clump fusing to the next one and forming a bigger cyst. So it seems that this loss of E-coherent and this disorganization of the cyst now makes the cells more motile and make them ability to now move in 3D and fuse to each other. Now, if we restore E-coherent expression by knocking down slag, as I show you in one of the slides, you can see now that this motility defect is completely restored. Now we can measure this by measuring the tracks of the cell, and what you can see is exactly what you see in the movies. Control cells don't move, the escape knockdown cysts move very much, and then the double knockdown when we rescue E-coherent, the phenotype is restored. So what are we trying to do now? So one thing that we did is try to see if this motility defect is also present in 2D. And I'm just going to show you one movie. It's very evident. You can see that the SCF knockdowns in 2D, they also migrate a lot faster. So what do we need to do now? Now we need, our imaging capabilities are going to help us a lot. What we need to do is to segment, quantitate, analyze a lot of these parameters to try to understand what's going on in here. We want to identify individual cells, measure the speed, the shape, their size, their position, and a lot of other parameters that will give us an idea of what's going on. And what we have been doing is we've been working with a group in Italy called AI for Life, and they use machine learning and different computer algorithms to segment cells and measure the different parameters. And this is a very early result of our attempts to segment. We're doing a lot better now, but what you can see this can give you is parameters for each individual cell. Where do they move? How do they move? Where are they in reference to one another, et cetera, speed and distance and all of these other things. So we're working with them And we're working on both segmenting in 2D and segmenting in 3D. And this is just one example. Let me see if it plays of what we're doing now. We're segmenting now in 2 colors. So now we can follow the nucleus and the membrane so we can see the cell divisions that you're going to be able to see in some of these cells as the cysts are forming over time. So the idea is that we can identify the cell, we can identify the nuclear, we can identify the cell divisions, we can map the positions and the distance from one cell to another, and this is going to give us a lot of information on how these cysts develop over time. So a quick summary of what we see in 3D. We see that the 3 members of the complex are important. We see something that I haven't talked much about, that there's loss of barrier function, there's reduced adhesion because we lose adherence, and we see this enhanced motility and fusion that we're very interested in trying to understand. So with that, I'm going to finish the presentation, and I'm going to thank the people that did the work. This work was originally initiated by a former student in the lab, Cecilia Wadia, who did all the initial study that I present from our previous publication. And now this work is being continued by Agustin, who's doing most of the 2D work and the coherent degradation. And then Mari is doing all the 3D cyst work and the image analysis. I would like to take just one more minute to thank the people that fund this research, mostly NIH, the General Institute of General Medical Sciences. And of course, I need to take a minute to thank Leica. For us, there's a before and after the Stellaris Award, so I cannot be more grateful of the support I got from Leica. So with that, I will be happy to take any questions.
All right. Well, thank you so much, Dr. Garcia-Mata, for your informative presentation. We will now start the live Q&A portion of the webinar. So to our audience, if you have a question that you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. All right, so let's get started. We've already got some great questions coming in here. And let's see, this first question asks, have you correlated your study with MMP2 and MMP9?
Thank you very much for the question. The short answer is we have not looked yet at MMP2 or MMP9 or any other metalloproteases, but it's definitely something that we're interested in, taking a look at in the future.
Great, thank you. All right, next question here asks, let's see. How do cells and tissue form an impermeable barrier, between the outside and the inside of tissues and organs?
Sorry, can you repeat the question?
Yes, I'll repeat the question here. The question asks, how do cells and tissues form an impermeable barrier between the outside and the inside of tissues and organs?
Yes, so as I briefly mentioned in the introduction, the barrier function of epithelial cells is mediated by these specialized junction called tight junctions. So that forms a very, very tight seal between 2 adjacent cells, and this seal will prevent the passage of solids and molecules through the cells and only allow for a very selective passage of some molecules and ions. So this is a multi-protein complex that has a very sophisticated way of being regulated, and we understand fairly well, but of course there's a lot of things that we still don't know about it.
Great, thank you. All right, next question here. In your MDCK model, I was a little surprised to not see some accumulation of beta catenin in the nucleus. Can you comment on this?
This is actually a great question and something that we're very interested in looking at. Actually, by immunofluorescence, you're right. don't see a lot of beta-catenin in the nucleus. And that may relate to the antibody, we're not sure. We have done some preliminary nuclear fractionation and with encouraging results. So we're pursuing these experiments to try to figure out whether there is a true increase in beta-catenin in the nucleus. I mean, clearly the inhibitor studies suggest that beta-catenin is implicated, but we haven't actually done the experiments to show conclusively that there's more beta-catenin in the nucleus.
Great, thank you. All right, next question here. This question starts with fantastic work, Rafa. This is Antoni's. I was wondering whether the SGEF effects that you see are directly to the E-cadherin complex, independent of scribble, especially since this is the membranous targeting of it that rescues it.
Let me go back, try to see the question again to make sure I'm answering correctly. Hold on just a second. I'm just gonna get back to it. Yeah, I mean, so there's a couple of questions in there. I still think that the complex is important. Forming the complex will target the activity to the right place. I don't think that the activity of SGEF is independent of the complex, but the complex is bringing the activity to the right place. The fact that silencing the other complexes doesn't have the same phenotype, I think that it has to do with the fact that the activity is mediated only by SGEF, and the partially impairing the complex by knocking out all the other two members does not phenocopy completely the loss of SGEF. So we're still sort of very interested in digging deeper in this. You know, what is exactly the role of each of the members, and also what is exactly the role of SGEF and Rho-G downstream? What are the effectors and what are the molecules that connect this complex with the regulation of EK hearing? So there's a lot of questions yet to be answered.
Yes, and we're getting so many great questions coming in from our audience, but unfortunately we're running out of time. So we have time for one more question here, and we'll wrap up after this one. This question asks, what are some of the tools we can use in the lab to image these processes in live cell and at high resolution?
Right, so at least the approach we're trying to take is to tag these the molecules that we're trying to image or we're trying to follow, mostly now trying to use knock-in. So we're trying to tag them at the endogenous level. So we want to try to stay away from overexpression and try to follow the regulation of these proteins when they are expressed at their normal levels in the cell. And we have been successful already in tagging EK hearing. So we're doing some experiments now with EK hearing tagged at the endogenous level. And the idea is sort of to build up and to tag more molecules so we can simultaneously track them in live cells, hopefully in 3D over time, and we can answer some of the questions that we still have.
Wonderful. All right. Okay, well, that brings us to the conclusion of our session today. So I want to thank you again, Dr. Garcia-Mata. Do you have any final comments for our audience before we go?
I just want to thank Leica and this webinar series for the opportunity to present our work, and also for the Stellaris Award. I'm very grateful for the support that I've been receiving from Leica for the past 2 years since we got the new microscope. So I just want to reiterate that and send my state my gratitude to the company.
Wonderful. Well, thank you so much. And thank you again for your time today and your important research. We'd also like to thank LabRoots and our sponsor, Leica Microsystems, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.